So this is the main screen for the PepArray Pro software. Here you'll find a description of what the software can do for you. And then on the right you'll see some of the things we're going to do. First, define an array format, design a panel of peptides, and then finally output the layout file. First thing to do is to define the layout. The first thing you'll see is up at the top left hand corner, the general description of the array. How many features are on the array, the amount of peptides that we have designed for the array, controls, things like that. We have a couple of options here for the array format. We have a 4 and a 5K chip. You can also see that there are some layout templates we can choose from. A phosphopeptide layout, an epitope layout, a kinase layout, and others. We're going to choose the phosphopeptide array layout for this example. You can see that the array layout information has been updated. Now with the total number of sites, the rows and columns, the number of QC controls that we put on every array, and those sites that are available to you, we're going to design the panel to fit within these available sites. Next, we're going to enter the source of the seed peptides. You can see that there are several potential sources, either from a file, from a database, from tiling across a protein. For this example, we're going to use peptides from the KEGG pathways. Listed here are the various pathways within the KEGG database. Next to the name of the pathway is the number of peptides that can bind to SH2 domain-containing proteins in the pathway we can use for detection on a phosphopeptide array. We're going to scroll down here and select the erbb signaling pathway as the source of peptides for our panel. You can see there are 614 peptides which can bind to proteins in this pathway that we can detect on our array. Here is the list of peptides which are going to be included on this array. Next to it is the sequence, the peptide sequence of the tyrosine binding domain, which is where we'll be able to detect that protein on the array. First, you can see that the array layout information has been updated with the 614 seed peptides that are now included within our panel. The next step will be to add some additional peptides to the panel that are derived from the original seed peptides. There are a few different options for creating derived peptides from the seed peptide sequences, for example, truncation, mutation, or alanine scanning. In this example, we're going to use mutation. Next, we need to specify exactly how we want the seed peptide sequences mutated, and we can put that information here. The position and then the mutated amino acid replacement. Here you can see the original seed peptide sequences all beginning with a tyrosine capable of being phosphorylated. In the derived peptide sequences, that tyrosine has been replaced with an alanine. These derived peptides will serve as a negative control without the potential of phosphorylation. We'll click this button to indicate that we want to add the derived peptide sequences to the array panel, and you can see over on the top left that they have been added to the total. We have a few options now. We can generate more seed peptides, we can generate some control peptides, or generate some more derived peptides. We're going to go ahead and make the panel with the peptides we have so far. Now we're going to enter the number of replicates of each of the peptide sequences on the array. For increasing the data confidence, let's put 3 replicates of each of the peptide sequences. We know that we have room left on the array, so let's use a replicate of 3. Next, we have the option to review the set of peptide sequences and number of replicates. Let's go ahead and move along. Next, we'll give this panel of peptide sequences a name, and we have the option to give it a description as well. And finally, we'll submit the panel. Here we see a list of the peptide sequence panels that we've created, in this case just the one, and next we can make and view the array. We'll need to give the array a name as we did the panel, and we also have the option to add a description here as well. We have a couple of options here as to how we want the array layout formatted. Now that the array layout has been created, we can view some of the statistics about the sequences that are going to be synthesized on this array. We can take a look at a graphical representation of some of the characteristics of this array, as well as the percentages of each of the amino acids that are going to be within the peptides that we synthesize on this array. And now it's time to export the array layout to an Excel spreadsheet. If we zoom in a little here, we can see the names of all of the peptides, both from the seed peptide and the derived peptide, as well as the control peptides, the QC controls that will be on the chip. And if we zoom all the way out, here you can see the entire layout of the 4K chip. This Excel file can be submitted to LC Sciences directly for the synthesis of your custom phosphopeptide array.